Good morning, students. Welcome back to today's the part of topic. So before to go the today's topic, I want to just recall once again the previous class double helix structure of DNA, as it is very important for the point of your board examination as well as CET and NEET examination point of view. I want to recall it because each one point in the DNA double helix structure you need to focus very much, because there are certain areas on which the CET questions would be expected. So let me start with one by one. So first of all, you need to know that who has proposed the double stranded structure of the DNA. So 3D structure of DNA model would be proposed or was proposed by the James D. Watson and the H.C. Crick in the year 1953. Then how did they propose? They proposed with the help of the X-ray diffraction data given by the Wilkins and Rosalind Franklin. So with the help of that, they are able to propose the double helix structure of the DNA. Then later in the year, for their wonderful contribution, altogether they have received the Nobel Prize in the year 1962. So let me recall then once again the double helical structure of the DNA one by one, the features or silent features of the DNA. So first point you need to concentrate while writing in the examination, who proposed this double helix structure of the DNA. The first point you need to concentrate, that is James D. Watson and Francis Crick proposed this double helix structure of the DNA. So this is the double helix structure of the DNA, where you can see each DNA molecule is made up of two polynucleotide strands. Each DNA molecule is made up of two polynucleotide strands, which are helically coiled in a right-handed manner along the common axis. Remember this point, along the common axis, so as to form a double helix structure of the DNA. In DNA, the two strands, the two polynucleotide strands, see, are in anti-parallel direction. This is also very important for the point of CET examination or NEET examination. The two strands in a DNA molecule, how they are whether parallel or anti-parallel in their polarity. It would be asked in the examination. You must know they are anti-parallel in nature. What is the meaning of anti-parallel? They are anti-parallel means the two strands run in opposite direction. That is means one strand has the 5' to 3' direction and other will have 3' to 5' direction. This 5' and 3' will be given based on the type of carbon atom of the pentose sugar which is found at the free end of the polynucleotide strand. So 5' means the fifth carbon atom of the pentose sugar where the phosphate is linked. 3' means the third carbon atom of the pentose sugar, that is deoxyribose sugar, where the hydroxyl group is found. Now come to the next point, as it says that the two polynucleotide strands, in the two polynucleotide strands, the sugar and phosphate molecules, the sugar, deoxyribose sugar and phosphate molecules are lying on the outer side, forming the sugar phosphate backbone. Where they are lying on the outer side, forming the sugar phosphate backbone. So like this, the number of nucleotides of each polynucleotide strand, they link together by forming a phosphodiester bond to form a polynucleotide chain. You have to remember here the bond which is going to link the adjacent nucleotide base pairs is phosphodiester bond, which is found between the fifth carbon atom of the one deoxyribose sugar of one nucleotide and the third carbon atom of the other deoxyribose sugar of the other nucleotide. Okay, so this is very important. Then next come to the third point, where are then nitrogen bases are present in the double stranded DNA. So nitrogen bases are attached to the right backbone at right angles to it. So nitrogen bases are lying on the inner side, and where they are attached to the backbone of the DNA, that is sugar phosphate backbone, at right angles to it. Okay, and now we'll focus on the next point. How does the two polynucleotide strands held together? The two polynucleotide strands held together by the hydrogen bonds formed between the nitrogen bases of the opposite strand. So it is very specific and complementary base pairing here. Complementary means the purines always pair with pyrimidines or vice versa. So like here you can say the purine base adenine always pairs with thymine. Okay, so or else it will be vice versa with two hydrogen bonds, and guanine always pairs with cytosine or vice versa with three hydrogen bonds. So this is called complementary and specific base pairing, so that there would be a certain CET questions to will be expected to predict that how the two polynucleotide strands are held together, either with the help of hydrogen bonds or with the help of phosphodiester bond or with the help of N-glycosidic bond. So that two polynucleotide strands are held together because of the hydrogen bonds formed between the nitrogen bases of the opposite strand. Then one more question will be asked here that is, why the DNA has got the uniform distance or uniform diameter or uniform thickness? Because of this complementary and specific base pairing mechanism, the DNA has got uniform diameter. So uniform diameter of DNA is because of its specific and complementary base pairing. Okay, then so each turn of the helix, which is going to be take at around for about 360 degrees Celsius, so that length would be always 3.4 nanometer, or if it is mentioned in angstrom unit, it would be of 34 angstrom unit. It is very, very important. So length of each turn in a DNA double helix will be 3.4 nanometer or 34 angstrom unit, and at each and every turn there will be approximately 10 base pairs. In each and every turn there will be approximately how many base pairs? 10 base pairs. With respect to this point, once again the CET questions are expected in the way that, so they may give you the number of base pairs and they may ask you to predict the number of turns, or else they may give you number of base pairs, they may ask you to find out the number of turns in a DNA double helix. Okay, so in this way it will be expected, and as there are 10 base pairs in each turn of the DNA, it is also known as pitch, then what will be the distance between the adjacent nucleotide base pairs? It will be 0.34 nanometer or 3.4 angstrom unit. So once again you remember, okay, so number of base pairs will be given in the CET examination, and it will be asked how many turns the DNA would be if the number of base pairs are in a length of the DNA is so and so. They may give and they may ask you to calculate the what is the number of turns of the DNA. Okay, so please focus on these areas, and in addition to that you must know how many number of bonds which would occur in the DNA. So there will be 3 different types of bond you come across in the DNA. That is, one is hydrogen bonds which would pair or link the adjacent, the opposite nitrogen bases of the opposite strand, and the phosphodiester bond which is going to link the adjacent nucleotides to form a polynucleotide chain or to form a nucleic acids. And another one will be the N-glycosidic bond, that is the bond which is going to link the nucleoside, that is nothing but the combination of the nitrogen base and the deoxyribose sugar. Let me highlight certain points in the double helix structure of the DNA on which the CET questions will be asked in the examination. So let me first focus it on the complementary base pair rule. So as the complementary base pair rule says that the purines, the total amount of purines will be always equal to the total amount of pyrimidines in a double stranded DNA molecule. That means if you know the sequence of nucleotides on one strand, one can predict, or you will be asked in the examination to predict the proper sequence of nucleotides on the opposite strand. So here let me take one example. Here I have written the sequence of nucleotides on one strand. So let me take what would be the sequence of nucleotides on this strand now. So as we all know that already, so adenine pairs with always T, so here it would be T. So thymine pairs with A, it will be A here. Like that, G pairs with C, C pairs with G, A pairs with T, T pairs with A, G pairs with C, C pairs with G. Like that, there would be some questions in your CET or NEET examination. So one strand, the sequence of one strand would be given, and it will be asked in the examination, find out the sequence of nucleotide bases on the opposite strand. Okay, so this is how you can easily predict the proper sequences. So this is the one pattern of asking the CET question. Okay, now so similarly there will be sometimes even questions which are related to Chargaff's base rule. Okay, so Chargaff's base rule is valid for only double stranded DNA. It would not be applicable for the single stranded DNA or else even for the RNA. So why it is not applicable? See, so Chargaff says that in a double stranded DNA, the total amount of purines will be always equal to the total amount of pyrimidines. That means the purines, as we know that the adenine and guanine, then pyrimidines are thymine and cytosine. So purine bases, that is A plus G, will be always equal to T plus C in a double stranded DNA. Okay, or else we can also write it as the ratio of A plus G and T plus C will be always equal to 1. Okay, so this is constant for the given DNA. For every DNA it will be constant. Okay, but see, the ratio of A plus T and G plus C will be constant only for the species. That means the ratio of A plus T means number of AT base pairs and GC base pairs are going to vary from species to species. Hence it would be helpful in identifying the DNA of the particular species. Okay, now with related to this, once again there would be the CET questions like that. Here I have mentioned one example here. So there will be some questions. How you know? See, in a double stranded DNA molecule, if the percentage of adenine is 15% and what would be the percentage of guanine? They will be asking the question like this. So how to work it out and find out the proper answer? See, as we all know that in an 100% DNA, all four the nitrogen bases together form an 100% DNA. That means A plus G plus C plus T all together form a 100% DNA. So here what is given here? Given is adenine. If adenine is 15%, then as we know that according to Chargaff's base rule, adenine will be always equal to thymine. When adenine is 15%, then thymine also will be 15%. So what we have to find out? We have to find out the G plus C ratio or the percentage. Now how to find out? Let's take here G plus C is equal to 100% minus the amount of A plus T. So 100% minus the amount of A plus T, as it is given already in the question. A is equal to 15%, as A is always equal to T, so A plus T would be 30%. So 100 minus 30% if you make, you will get 70%. So divide by 2. So as you have to find out the ratio for amount or percentage for both G and C, so divide it by 2 and you will get G is equal to 35% and C is equal to 35%. This is how many more questions will be there in the CET examination. And depending on, not only that, depending on even the denaturation and melting point of the DNA, also there would be certain CET questions. Okay, so what is denaturation? Denaturation is nothing but splitting of this double stranded DNA molecule into single stranded. When it will get split, it will be getting splitted when you subject it for the high temperature or when you heat it. Okay, how it would be able to get into separated into single stranded? By breaking off its hydrogen bonds. As we all know that hydrogen bond is a weak bond. Okay, as here two types of bases, purine bases and pyrimidine bases, and A always pairs with T with the two hydrogen bond. Okay, so it will be weaker when considered to the G pairs with the 3 hydrogen bonds with the C. Okay, so listen here once again. So AT base pairs are weak when considered to GC base pairs, because GC base pairs will have 3 hydrogen bonds, AT base pairs will have 2 hydrogen. That is why, so based on that, the melting point would be calculated. So more is the GC base pairs, then more will be a melting point of the DNA. So less is the GC base pairs or more is the AT base pairs, more will be the, less will be the melting point of the DNA. So like that, generally it would be, taken the melting point of the prokaryotes, it will be always greater than 1. Melting point of the eukaryotes, it will be less than 1. Okay, so it indicates that the prokaryotes have got more number of GC base pairs when compared to the eukaryotes. Okay, so that means denominator value is more in case of prokaryotes, where the numerator value is more in back. So we shall move on now, the different forms of DNA. So DNA is commonly found in the classic form in all the living organisms. So that is nothing but the Watson and Crick DNA. So that is simply known as bDNA, as the DNA is going to follow all the criteria of the Watson and Crick model. So the Watson and Crick DNA is nothing but the bDNA. So apart from that, at certain conditions, it is also found that DNA is going to be found in other forms also. So the other forms vary greatly with the structural wise. So how they vary the structural wise? So they all vary, the different forms vary with respect to the number of base pairs per turn and the mode of their coiling. Okay, so hence there are how many types? A DNA, B DNA, C DNA, D DNA, E and Z DNA. So totally there are 6 types of DNA have been found here. So in case of A DNA, let us know what is the mode of coiling here. It is right-handed coiling, and number of base pairs present per turn, there are 11 base pairs. Okay, in case of B, as I have already mentioned, B DNA is nothing but the Watson and Crick model of DNA. So hence it is having the right-handed coiling, and the number of base pairs per turn will be usually 10 base pairs. And in C DNA, it is again right-handed coiling, and the number of base pairs per turn will be approximately 9 base pairs. And in case of D DNA, once again it is right-handed coiling, the number of base pairs are 8 per turn. In eDNA, right-handed coiling, number of base pairs are approximately 7 base pairs. Okay, so these different forms of DNA you can easily remember here, as I have mentioned here. They are all having the descending order. The number of base pairs are arranged in descending order. So in case of A there are 10, in case of B 10, in case of C approximately 9, D 8, in E 7 approximately base pairs per turn. And in case of Z it is totally different when compared to the all other forms. So Z is having the left-handed coiling, and in addition to that, see, there are 12 base pairs in each turn. Okay, so these are the different forms of DNA where you need to remember for the point of your CET examination as well as for the NEET examination. Now we shall move on to the another form of DNA which you can also come across in the recombinant DNA technology topic. So that is called palindromic DNA. So what is that? The palindromic DNA. So what is the meaning of term palindrome? Palindrome is a term which reads the same letters when you read from both forward and backward direction, like Gadag, which reads same letters in both forward and backward. So similarly Malayalam, okay, which also reads the same letters from both forward and backward direction. So such type of DNA is called palindromic DNA. So what is the meaning of palindromic DNA? Is a small segment of DNA in which the sequences read same in both the strands, where here I have mentioned one example. In one strand, that is in 3' to 5' direction, where you can read the nucleotide base pairs, they are GAATTC. So again in the opposite strand you can see in the same direction, 3' to 5' direction, they read again same GAATTC. So such segment of DNA is called palindromic DNA, which is having very important importance in recombinant DNA technology. We'll focus it on the later classes. Now in addition to that, structurally we would come across the two types of DNA, that is linear DNA and circular DNA. So one is the linear DNA. Linear DNA, all the eukaryotic DNA is double-stranded and it is linear. Whereas in case of prokaryotes as well as in case of cell organelles, where you can find the double-stranded DNA but it is circular. So with respect to structural wise, we can find two types of DNA. With respect to the base pairs and mode of coiling, we are going to find the total 6 forms of DNA. So this is about the forms of DNA. Now we shall move on to the very interesting topic, that is called central dogma. So what is this central dogma? Central dogma is the concept which is proposed by Francis Crick, the one who proposed the double helix model of the DNA. Okay, so this concept is proposed by Francis Crick in the year 1958. So it states that genetic information flow in one direction, that is unidirectional flow of genetic information, that is from DNA to RNA, from RNA to protein. From DNA to RNA, from RNA to protein. Then protein expresses the phenotype. So this is called central dogma. So central dogma means always the genetic information flow in one direction. That is how you know. So you all here, you can wonder here how we are all have got a certain resemblance to our parents or our grandparents. Here is the answer, because here DNA has got the property of replication. So whatever the DNA which you have inherited, that is brought from your parents. So because of the property of replication. So which DNA which is inherited to you, it has the another property, it is getting transcribed to bring the same genetic information from your parents to you and express it in the form of phenotype, that is in the form of protein. So protein expresses the same resembling feature among you. So this is called the central dogma. So where the flow of genetic information occurs always in one direction, that is from DNA to RNA, that is nothing but the mRNA through the transcription mechanism. Okay, then from RNA to protein, once again through the translation mechanism. So protein is nothing but the a polypeptide chain of amino acid, so that is expressed here. So this is nothing but the expression of the particular gene among the individuals, so which resembling, whether the individuals they are resembling or they are varying from the parents, that depends upon the genes which are you are going to be inherited. If you are inherited the same gene, that you are going to inherit the same phenotype, or you are going to express the same phenotype. If you have difference when compared to the parents, that is because of recombination or crossing over which is taking place during the meiosis of the type of cell division. Now we shall move on to the another concept where the Temin and Baltimore, they brought forward some exception to the central dogma. So what is that exception? Here H. Temin and D. Baltimore, they brought forward that one exception to this central dogma of reverse, that is called central dogma reverse. Why? What is the meaning of this? See, central dogma says that the flow of genetic information in only one direction, that is from always DNA to RNA, from RNA to protein. But in case of certain tumor viruses, means cancer causing viruses, where they have the RNA as their genetic material, how it would be possible to bring the genetic information in one way? No, it is not possible. So hence it is mentioned here as central dogma reverse, because RNA has to get into converted into DNA. So such DNA which is getting formed by the RNA is called complementary DNA. So C stands for here complementary. So that cDNA is nothing but the complementary DNA which is replicated from the genetic material, that is RNA, which is present in the RNA viruses. So one of the typical example if we can take, that will be HIV. HIV, the human immunodeficiency virus, which is causing the AIDS, which is having RNA as their genetic material. So in such cases the genetic information will flow in two directions, that you can see here, the from DNA to RNA, okay, and RNA to protein. It is the central dogma of molecular biology. If it is replicating in the reverse direction, then it is called central dogma reverse. So it would be expressed as the RNA as the genetic material which is present in the HIV or other retroviruses, okay, that is going to be replicated to form a cDNA. Then further cDNA is going to be transcribed into mRNA, and mRNA will be expressed as the particular protein, and protein. Okay, now we shall move on to the packaging of double helix of DNA. As we all know that DNA is found as a long polymer, so means long chain of polynucleotide. Okay, so how it would be measured in lengthwise? If we want to measure, so we have to multiply the number of base pairs with that of the distance of adjacent base pairs. So if you take a typical mammalian cell as an example, the human cell, okay, in a human somatic cell there would be 6.6 into 10 to the power of 9 base pairs. So if it is multiplied by a distance between the consecutive base pairs, that would be 0.34 nanometer, that is into multiplied by 9. So you will be getting approximately 2.2 meters long DNA. So here the question arises, how this such a long DNA being 2.2 meter, how it is possible to get resided or enclosed within such a small nucleus? So dimension of the nucleus in an eukaryotic cell is 6 micrometers. So it will be there must be certain special mechanisms which would have involved here to get the packed this such a long polynucleotide chain, that is DNA, to reside in a such a small nucleus. So let us see that now how the packaging would take place in the prokaryotic cell and in case of eukaryotic cell separately. In case of prokaryotic cell, if you take in the prokaryotic cell, there is about 1.36 millimeter of DNA. So such 1.36 millimeter of DNA, how it is enclosed in a prokaryote. So let us take the prokaryotic cell, E. coli, a typical example, E. coli bacteria. So as in case of prokaryotic cell, there is no well-defined nucleus. That doesn't mean that the DNA is scattered throughout the cell. So actually the DNA, it is being negatively charged, being associated with proteins and forms a highly looped structure called nucleoid and held in a particular region. So in case of prokaryotes, the packaging mechanism is very simple. So how it would be? In case of prokaryotes, the typical E. coli bacteria, which has got around 1.36 millimeter of DNA, which is being negatively charged, associated with proteins which are positively charged, and forms a highly looped structure called nucleoid here. Okay, and that is not freely scattered throughout the cytoplasm, which is held restricted to the one particular region. Now we shall move on to the similar packaging of DNA in case of eukaryotes. In case of eukaryotes, as a typical mammalian cell, what I have taken in the earlier, so that is the 6.6 into 10 to the power of 9 base pairs of the typical mammalian human DNA would be multiplied with the 0.34 into 10 to the power of minus 9 meter. So you will be getting approximately 2.2 millimeter DNA. So 2.2 meter DNA, how it would be possible to held in a such a small nucleus. So what is the dimension of the nucleus I mentioned? 6 micrometer. 6 micrometer it is. Okay, now there must be a complex mechanism of what you can say packaging is involved. So what is that complex mechanism of packaging of DNA in case of eukaryotes? So in case of eukaryotes, actually there are set of positively charged proteins called histone proteins being associated with negatively charged DNA. They form a particular structure called nucleosome. So how it is going to be formed? See, what are histone proteins? First let me tell about that. So histone proteins are the basic rich in basic amino acid, and these basic amino acids are usually lysines and arginines, which have got the positive charges in their side chains. So because of that, the histone proteins have got the positive charge. So this histone proteins, there are 5 different types of histone proteins get involved into the packaging of double helix of eukaryotes. They are H1, H2A, H2B, H3 and H4 types, out of which the 4 different types, so H2A, H2B, H3 and H4 are going to occur in pairs to form the 8 histones or the core of 8 histones called histone octamer. It is also known as a new body or new body. Okay, so this gets positively charged. So this is being positively charged, now attracts the negatively charged DNA. So when positively charged histone octamer come close to the negatively charged DNA, so DNA gets wrapped around the histone octamer by its 2 turn about 200 base pair long helix to form a structure called nucleosome. So nucleosome is the fundamental unit or repeating unit of a chromatin or chromosomes. Each nucleosome will have a diameter of about 10 nanometer. So, and there will be several nucleosomes would get formed, and those nucleosomes are linked together by the help of linker DNA, and each one nucleosome is going to be held together with the help of histone octamer. So that is with the help of H1 histone. So H1 histone is going to involve only in the holding the histone octamer with the wrapped DNA around it. Now the number of nucleosomes are going to found linked together with the help of linker DNA, and such a nucleosome structure under an electron microscope, it is going to give an appearance of beads on string under an electron microscope. So nucleosomes appear as beads on string. So such chromatin which gets packed once again and recoiled to form a chromatin fibers, so which further coils and condenses to form a highly condensed rod shaped structure called chromosomes during the metaphase of the cell division. So let me show you how that the structure of the nucleosome. So this is, you can find here, this is the histone octamer which is formed by the 8 histone molecules. I told there are 5 different types of histone molecules are involved in the packaging of DNA double helix in case of eukaryotes. They are, one is H1 and other 4 types are H2A, H2B, H3 and H4 type. So these 4 different types of histone proteins occur in pairs. Okay, so all together they are going to form 8 histones, and 8 histones, when they occur together, it is called core of histones, or you can say histone octamer. So this histone octamer getting positively charged, because of that the lysine and arginine base pairs, okay, amino acids, okay, so which is going to get the positive charge here. So this positively charged histone octamer attracts the negatively charged DNA. When it attracts, the DNA gets coil around the histone octamer by its 2 turn about 200 base pair long, okay, and then such a structure is going to be held by the help of H1 histone, so as to form a nucleosome. So what is a nucleosome? I have already mentioned nucleosome is the fundamental unit of chromatin. Okay, so this is the one diagram which is given in your NCERT textbook, which is asked in the examination to draw a neat label diagram of a nucleosome. So you must have to practice this diagram. Okay, so in this you can say the ball like structure which you are going to see, this is indicating the histone octamer which is formed by 8 histone or core of histone molecules, where the 8 histone proteins are involved here, and the 2 turns of DNA, this is the entry of the DNA around the histone octamer, this is leaving end of the DNA, okay, histone octamer. So you can see here each nucleosome is held in position with the help of H1 histone. So this is the nucleosome structure, and I have one more slide here to show how these nucleosomes are linked together. So this is one nucleosome, this is another nucleosome. So several nucleosomes are going to link together to form a number of or polynucleosome structure, which is going to appear as beads on string under electron microscope. So you can see here the adjacent nucleosomes are going to be linked together through the help of linker DNA, and each nucleosome is held in a position with the help of H1 histone, and the dimension of each nucleosome is 10 nanometer. In chromatin fibers, you can find the 2 types of chromatin. So one is euchromatin and another is heterochromatin. What is this euchromatin and heterochromatin? When the chromatin fibers are stained with nucleic acid stains, so there are regions which take the darker stain and there are regions which take the lighter stain. The region which takes lighter stain is called euchromatin. The region which takes darker stain is called heterochromatin. It takes lighter stain because where the DNA is loosely packed here, hence it takes the lighter stain. Heterochromatin takes the darker stain because here the DNA is densely packed. Okay, now with respect to that, even they have other differences, that is transcriptionally this euchromatin will be more active and even it is early replicating, whereas heterochromatin is transcriptionally inactive and it is late